our DiAGSure HPV-16 high-risk genotype detection kit. I'll just open the kit. It's the kit insert. As evident, the kit contains one master mix, which is called WR-Tachman master mix, one primer probe mix, one positive control, one negative control, and one DNA dilution buffer. These are basically the kit components. I'll test 3 samples. So for 3 DNA, one positive control, one negative control. So I'll prepare master mix for 5 samples. So per sample, 13 microliter of this WR-Tachman master mix is required. Therefore, I'll take 5 into 13, that is 65 microliter of this WR-Tachman master mix. I've set the pipette at 65. I'll pipe it out 65 microliter of this WR-Tachman master mix. Put it in then 1.5 ml centrifuge tube. Primer probe mix, the next reagent. Per sample, 2 microliter of primer probe mix is required. Therefore, for 5 samples, 2 into 5, that is 10 microliter of this primer probe mix will be added. Tap mix the solution a bit. Next we will allocate 15 microliter of this master mix into the respective wells. Next we will add the sample DNA, 10 microliter each, into the respective wells. And finally the controls. First the negative control. And lastly the positive control. Next we will seal the plate. Put the plate in a PCR machine. Set up the program this way. Select the channels FAM, HEX, ROX and Sci5. FAM is for HPV-16, HEX is for HPV-18, ROX is for HPV others, and Sci5 is your internal control. We'll label the positive and negative controls. And then start the run. This is how the positive control amplification looks like. Amplification in the FAM channel symbolizes HPV-16 amplification. Amplification in the HEX channel or WIC channel indicates HPV-18 amplification. ROX channel amplifications would indicate HPV other high-risk genotype amplification, and amplification in Cy5 channel would indicate internal control or RNSP amplification.